Very good morning, class 11th. I hope that you all are doing really well. So, in today's lecture, we are going to talk about the oxidative phosphorylation. If you just recall your previous lecture, in our previous lecture, we talked about two types of phosphorylation, that is substrate as well as oxidative phosphorylation. In today's lecture, I am going to tell you about that how from NADH2 and FADH2 we are going to get our ATP molecules. We have now we understood that how electrons they are getting transported in the electron transport chain. Thoroughly we have done it, but now how ATP is synthesized, that we are left with. So today we will talk about oxidative phosphorylation, and one idea you have in your mind that oxidative phosphorylation means ATP is going to synthesize from NADH2 and FADH2 molecules, means ATP is going to synthesize indirectly. So in oxidative phosphorylation, if I ask you that it is what it is about ATP synthesis in the mitochondria. As you know, all of you are aware that mitochondria is our powerhouse of the cell. So there is some energy released during the etc, during the transfer of electrons, right? Energy is released during transfer of electrons from higher energy level to lower energy level. And once the electrons they move from higher energy level to lower energy level, they release energy, and this energy is released during ETS, that is electron transport system. It is utilized in synthesizing ATP with the help of our complex number 5, and this complex number 5 is known as ATP synthase complex. What is so special about this complex? This ATP synthase complex, it contains two particles, that is F0 and F1 particles. So if this is your outer membrane, this is our intermembrane space, or it is the space between the outer and inner membrane. This is intermembrane space, and this foldings, these are our crista, and this membrane is inner membrane. Now in the inner membrane we have our channel protein, that is your F0 and F1, right? So this one is our F0, and there is the stalk, or this is, you can say, body of F0 F1, and this the extrinsic protein which is present in the matrix, that is our F1 protein. So this is inner membrane, and this is the intermembrane space. Now see, during ETS we discussed that protons, the moment the inner electrons they are transferring from higher energy level to lower energy level, protons they are jumping in, right? Protons they are pumping inside the space, intermembrane space, from the matrix, that we have learned. Now once our protons they will move here in from matrix to the intermembrane space, now there is a gradient is formed, that in the intermembrane space there is a high concentration of protons as compared to the matrix. In matrix there is the low concentration of protons. And how to break down this gradient now? If these protons they have to move back to matrix, because there is a movement always from higher concentration to lower concentration. So if these protons now they have to move back to the matrix, they need to pass through the channels, they need to pass through the gate, and these are our F0 F1 proteins. F0 is our intrinsic protein which is embedded inside the inner membrane of mitochondria, and F1 is the extrinsic protein which is present towards the matrix. Now protons, there is only one way for the protons, that they can only move from F0 to F1 proteins if they want to go back to the matrix. And actually they need to go back because there is a high concentration and there is low concentration. So movement will be from high to the low concentration. So F0, F1, they act as a channel. Now, once the movement of proton, the proton, it moves from intermembrane space and it reaches to the F0. F0 helpful is act as a diffusion, facilitated diffusion in the movement. Now it is coming with the full of energy. The moment it reaches the F1, F1 इसकी एनर्जी यूज़ करता है and that energy is utilized for combining the ADP with the inorganic phosphate. The moment this proton reaches to F1, the energy which is it is releasing, that energy is utilized here to combine the ADP plus inorganic phosphate, and one ATP is going to synthesize here because of the presence of enzyme, that is ATPase in the F1, and then this proton is pumped into the matrix. This proton is thrown into the matrix. So how is it moving? It is moving from intermembrane space to the F1 to the matrix through the F0 F1 channels. So F0 and F1 here, they act as a channel. The pair of protons, so from one NAD, so pair of protons, one proton will be released, one ATP will synthesize, second proton will move, second ATP will synthesize, third proton, pair of proton will move, then third ATP will synthesize. So in this way, the movement when electrons they are passing from higher to lower energy level, जो हमारी इलेक्ट्रॉन ट्रांसपोर्ट चेन है उसमें जैसे FMN, FES, UQ, UQH2, there are multiple proteins. So when electrons they are passing through these proteins, they are releasing energy, and when they are releasing energy, that energy is that energy is using to pump out the protons. Now it means when they have to go back, every time they have to return to back, they have to move through the F0 and F1. So with the one pair of movement, one proton when they will move, one ATP will synthesize. So second will move, second ATP will synthesize. Third pair will move, then third ATP will be synthesized, right? So in this way, ATP molecules, they are synthesized from the oxidation of one NADH2, because on the oxidation of NADH2, protons they are going to make a gradient in the intermembrane space, and breaking down of that gradient, जब उस gradient को तोड़ा जाता है, जब ये move करते हैं from higher to lower, only then ATP is synthesized. So when FADH2 passes the electron, it releases the energy at two places, and when it releases the energy, protons pumped in, and when NADH2 passes the electrons, energy is released at three places and three protons pumped in. Now you can understand that in during FADH2 oxidation, two ATPs, that is why we are saying, because two protons they are pumping in, so two protons means they have to go back to the matrix, so two ATPs they are synthesized. And during NADPH2 oxidation, three places where energy is released, and three places where protons they are coming in. Once they go back to the matrix, 3 ATP they can be synthesized. So the moment these protons they move back to matrix, so here 3 protons they were pumping in, so 3 have to move back, so 3 ATP are formed. Here 2 protons they were coming into the intermembrane space, they have to move back to matrix, so how many ATP? 2 ATP. That is why we are saying that here we are getting our ATPs from the oxidation of NADH2 and FADH2 by breaking down of proton gradient. This line is very 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 important for you. अगर gradient form नहीं होगा तो there is no need to go back into the matrix. So gradient बहुत important है for the formation of ATP. So complex 5, till now we are very much aware about the 4 complexes. If you have not read about ETS right now, so please it is the time that go through that go to that topic, go through that topic and read it. So complex 5, it consists of two major components, that is F1 and F0. F1, how does it look like? It is the structure of this ATP synthesis complex is like this, right? So F1 is a headpiece. Basically, it is a peripheral membrane protein. It is present towards the outer side and it acts as a channel through which protons they're going to cross the membrane. Which membrane? Here we are talking about which membrane? That is your inner membrane. So always we need to understand that two protons, the pair of protons, there is always a pair of protons pass through F1 from the inner membrane and they move to the matrix, and only then one molecule of ATP synthesized. If you are mentioning, always try to, they are pair of protons which are moving back to the matrix and they passing through the inner membrane. So now oxidation of one molecule of NADH2. Oxidation, when NADH2 undergoes oxidation, it releases energy at 3 places, due to which 3 protons, 3 pair of protons, they come into the intermembrane space, and once they go back to matrix, because they have to cross the inner membrane, so that is why 3 molecules of ATP are synthesized. And during the oxidation of one molecule of FADH2, energy is releases at 2 places. 2 places means 2 pair of protons, they are coming into the intermembrane space. Once they have to go back to the matrix, so they are going to give rise to 2 molecules of ATP. So here let's count now our total number of what we got from our respiration. Glycolysis. In glycolysis we talked about, and here is the intermediate step, that is the pyruvic acid. From pyruvic acid we formed our acetyl coenzyme A, CO2 was released there, and Krebs cycle. We will not talk about ETS here because ETS is not directly involved in ATP synthesis. So see, glycolysis, we got how much ATP from glycolysis? Directly 2 ATP they were synthesized there in the glycolysis. And in decarboxylation of pyruvic acid, there was no ATP, it was NADH2 which was synthesized. In Krebs cycle also we got our, see if you recall Krebs cycle, that we got our 1 GTP in 1 pyruvic acid, but the condition was that we have to use 2 pyruvic acid for the Krebs cycle, so we got our 2 ATP. So here in if we talk about FADH2 and NADH2, then in glycolysis the NADH2 which were produced, that were 2, right? So 2 and 1 NADH2 is equal to 3 ATP, so 2 is to 3, that is 6. In pyruvic acid also, 1 pyruvic acid was giving us 1 NADH2, but we already we have to talk about 2 pyruvic acid, so 2 NADH2, again here 6 ATP. In Krebs cycle we were having our 3, right? If you recall, there were 3 NADH2, because we are not going to include this step in Krebs cycle now, so individually only 3, but 2 pyruvic acid, if 2 pyruvic acid are forming acetyl coenzyme A, then 6 NADH2 as well as 2 FADH2, so total was the 18 ATP and 4 ATP. So if let's grand total now, 6 plus 2, 8 ATP. Here 6 ATP in our pyruvic acid, and in Krebs cycle we got our 18 plus 4 as well as plus 2. So here that is 24 ATP. Now if you calculated the total amount of ATP which we got during respiration, that is our 38 ATP molecules in aerobic respiration. But usually we talk about only 36 ATP molecule. So what about the two? See, if the question is about the prokaryotes, if question to you is asked about prokaryotes, then no problem with that. 38 ATP molecules are produced from one molecule of glucose during aerobic respiration, not at all a problem. But see, in eukaryotes, because they are little complex, and this step was happening in our cytoplasm, and now there was the movement of pyruvic acid from cytoplasm to the mitochondria. If you recall that, then there was a movement from cytoplasm to mitochondria. So two in eukaryotes, I'm talking about in eukaryotes, two molecules of ATP are utilized there for transporting two molecules of how many? Two NADH2, two NADH2 during glycolysis from cytoplasm to the mitochondria. So if we now cancel our this 2 ATP, then how much is left here? Here we will left only with the 6 ATP in case of eukaryote, and the total amount will be then 36 ATP, because these 2 ATP you have used for the transportation of this NADH2 from cytoplasm into the mitochondria. So that the net gain of ATP in eukaryotes will be 36 molecules of ATP. But if the question they asked about prokaryotes, then it will be 38. But in eukaryotes, because we are utilizing two molecules, so the total net gain will be 36 molecule of ATP. In the last, we are only now left with our two topics, that is the one common pathway for proteins, fats and carbohydrates, as well as the RQ. So in my next lecture, I'm going to talk about the last topic of our respiration, that is about the respiratory quotient.